For more than a century, evolutionary biology was largely a science of the past. To understand how species changed, researchers had to rely on the static, fragmented evidence left behind in the fossil record. The primary frustration was one of time. Because the adaptation of complex organisms plays out over millions of years, scientists couldn't watch it happen in real time, nor could they run controlled, repeatable experiments to see how the process actually works. In 1988, a biologist named Richard Lenski set out to fix that problem. He launched an ambitious initiative that helped define the modern field of experimental evolution. By pulling the study of adaptation out of ancient geological strata and putting it directly onto the laboratory bench, researchers could finally ask and answer entirely new questions about how biology changes. Lenski began his experiment with a single asexually reproducing clone of the bacteria Escherichia coli. That single clone was then divided, allowing Lenski to start with 12 entirely separate, genetically identical bacteria populations. Each of these 12 populations was placed under severe environmental constraints. Every single day, all 12 groups were fed the exact same, highly limited diet of glucose. This exact duplication sets up the core question of the long-term evolution experiment. If you start with 12 identical genomes and place them in 12 identical environments, will they all evolve the exact same way, or will random chance steer them down different paths? It is a direct physical test of the tension at the heart of biology, the chaotic, random nature of genetic mutations on one side, and the highly specific, deterministic pressure of natural selection on the other. To measure how these bacteria changed over time, Lenski developed a rigorous archiving system. Every 500 generations, researchers extract samples from all 12 flasks and store them in freezers set to -80 degrees Celsius. Plunging the bacteria to this temperature puts them into a state of suspended animation. It creates a living fossil record that can be thawed out and revived exactly as it was. This allows researchers to perform a literal head-to-head competition across time. They can thaw out an ancestor from generation 0, place it in a flask, and force it to compete for limited glucose against its own distant descendant. To see who wins the battle, researchers plate the mixed cultures and use a neutral color marker built into the strains. By simply counting the distinct red and white colonies, they can calculate exactly which population reproduced faster. This time travel methodology gave biologists something they never had before: an objective, purely mathematical measurement to prove that an organism has adapted to its environment. Once adaptation could be precisely calculated, the researchers wanted to map the speed of these changes. They wanted to know if fitness climbed upward in a slow, steady progression. This chart plots the mean fitness of the bacteria relative to their ancestor over the first 2,000 generations. You can clearly see an overarching upward trend as the bacteria adapt, but the progression itself is highly irregular. Zooming in on one population, a distinct staircase pattern emerges. The flat plateaus represent hundreds of generations without major changes. Sudden vertical jumps occur when a beneficial mutation randomly appears and rapidly outcompletes the rest of the flask. The staircase perfectly visualizes punctuated dynamics. It proves that phenotypic evolution, the actual physical changes in an organism, does not require a slow, imperceptible crawl. It can happen in rapid, concentrated bursts. Looking across all 12 independent populations gives us a clear answer to the experiments found in question. In terms of overall survival and reproduction, adaptation was highly repeatable. Over the first 2,000 generations, every single one of the 12 populations increased their relative fitness by approximately 37%. Faced with the exact same glucose-limited environment, they all reliably found similar solutions. But underneath that similar outcome lies a chaotic reality. Because the underlying mutations arise by chance, how the bacteria achieved that 37% increase varied wildly. At the genetic level, each flask accumulated a completely unique set of mutations. The most extreme example of this divergence occurred much later in the project. One single population randomly acquired a novel ability to consume a different molecule, citrate, from the culture medium, a trait that the other 11 lines never developed. These two separate outcomes revealed the dual nature of evolution. Natural selection acts as a predictable, unifying force, driving organisms to adapt. but it constantly runs on an inherently unpredictable engine of random genetic mutations. The long-term evolution experiment did not stop at generation 2000. The daily transfers of bacteria have continued uninterrupted for decades, recently surpassing 80,000 generations. Richard Lenski's methodology proved that evolutionary biology is a living, mechanical reality that can be directly observed and quantified in real time. By freezing generations and competing them against their descendants, this living time machine demonstrates that biological life is never a finished product, but an active, continuous process of adaptation that we can finally watch unfold.